Welcome back to the deep dive. Our whole goal here is taking these big, complicated subjects, today it's human genetics, and really boiling them down to what you actually need to know. We want to give you that shortcut to feeling genuinely informed. And today, we're tackling classical Mendelian inheritance. We're going beyond, the basic pea plant stuff you might remember, and looking at the real clinical tools, the specific language, and even the, well, sometimes tricky math used to predict risk for inherited diseases.
That's exactly right. We're drawing from some pretty high-level academic notes on human genetics today. The aim for this deep dive is real understanding, getting zygosity, being able to interpret those 5 core inheritance patterns, knowing how to read pedigree chart, and critically calculating that recurrence risk.
And you can't talk about this without mentioning Gregor Mendel, right?
Oh, yeah.
66, those pea plants.
Yeah.
He basically laid out the rules showing traits are passed down in predictable ways, not just blended together. His work kind of got lost for a bit, then rediscovered around 1900, and bam, it's the foundation for modern genetics.
Absolutely. So to really get a grip on this, we need to start with the language. And a core concept is zygosity. It sounds technical, but it just means the combination of alleles, think gene variants that you have at a specific spot, or locus on a chromosome. So if your 2 alleles at that spot are identical, like 2 capital A's or 2 lowercase a's, you're homozygous. Simple enough. If you've got one of each, you're heterozygous.
Okay, homozygous, heterozygous.
Got it. But here's a term that's really crucial, especially in clinical genetics: compound heterozygous.
Ah, okay, compound heterozygous. That sounds more like something you'd see in a patient report than just what does that actually mean for someone?
It means you have 2 different bad alleles, pathogenic alleles, in the same gene. So you got one specific mutation from mom and a different specific mutation from dad, both in that same gene. Technically, you're heterozygous, but it's important to know the errors are distinct.
That makes sense. So the type of error matters, not just that there is one.
Yeah.
Okay, so if zygosity tells us what allele someone has, how do dominant and recessive tell us what we actually see?
Right. That's about expression. If an allele is dominant, just having one copy, so being heterozygous AA, is enough for the trait or disease to show up. But if it's recessive, you need 2 copies of that allele, the A state, for the trait to be expressed. If you're heterozygous AA for a recessive condition, you're typically an unaffected carrier. You carry the gene variant, but don't show the symptoms.
And the tool we all learned in school to visualize this is the Punnett square. Assuming folks remember the basics, the key clinical point is that AAKA cross, right? 2 heterozygous parents.
Exactly. That cross gives you that predictable 25% chance 1 in 4 of the child being A homozygous recessive. And that 25% is the number we really focus on for recurrence risk in recessive diseases.
Okay, let's make this real. Let's use that language, especially compound heterozygous, and look at this case study from the source material. It's about Emily, a 5-month-old baby. She had some classic worrying symptoms. Lots of respiratory infections, wasn't growing well, what they call failure to thrive, and these frequent, greasy, bad-smelling stools.
Yeah, those symptoms together, especially the stool description, strongly suggest a problem with absorbing nutrients. And the test that really nails the diagnosis here is the sweat test. Emily's chloride levels were sky high, averaging 102 millimol. Normal is usually under 60. That sweat test result confirms cystic fibrosis, or CF. It's an autosomal recessive disease, meaning it's on the non-sex chromosomes, and you need 2 copies of the faulty gene. It's actually surprisingly common, particularly in people of European descent, about 1 in 2,500 newborns.
So what's actually going wrong in the body with CF? Why does this faulty gene cause things like thick mucus?
It's all down to a protein called CFTR, which acts as a chloride channel. When it doesn't work right, the balance of salt and water across cell membranes gets messed up, especially in the lungs and pancreas. Basically, salt movement is impaired, water follows salt, and if salt can't move out properly, water stays in the cells, making the mucus on the outside thick and sticky.
And Emily's case is a perfect example of being compound heterozygous, isn't it?
Exactly. Her genetic testing didn't just show she has 2 CF mutations. It specified which 2 different ones she had. One was a specific deletion, the PF50A-DEL from her mom. The other was a different type of error, a substitution called P-Li5501-ASP from her dad.
So 2 different bad copies of the CFTR gene. And crucially, both her parents are healthy.
Correct. They're both carriers, heterozygous AO, which confirms that autosomal recessive inheritance pattern. They each passed on 1 faulty copy to Emily.
It really drives home that these aren't just abstract ratios. It's about real families, real risks. And tracking that risk through a family requires a specific tool, the pedigree. right? The
Yeah.
pedigree. It's basically a standardized family tree.
Mhm.
You need to know the symbols: square for male, circle for female. Filled in means affected by the trait. And there's usually an arrow pointing to the proband. That's the person who brought the family to genetic attention. So Emily in this case.
Mhm. And seeing that visual pattern helps us figure out how the trait is passed down and, importantly, calculate the odds for future generations. Being able to look at a pedigree and quickly figure out the likely mode of inheritance, that's a key skill. There are five main Mendelian patterns we need to recognize: autosomal recessive, AR, autosomal dominant, AD, X-linked recessive, XLR, X-linked dominant, XLD, and mitochondrial.
Okay, let's break those down. Starting with autosomal, meaning the genes on chromosomes 1 to 22, so it generally affects males and females similarly. We just talked about autosomal recessive, AR with CF. Typically, parents are unaffected carriers. There's that 25% risk per child. And on a pedigree, you often see a horizontal pattern. Only siblings in one generation are affected, it seems, to skip generations.
Right. Contrast that with autosomal dominant, AD. Here, just one copy of the mutated allele is enough. So affected people usually have an affected parent. This gives a very clear vertical pattern. On the pedigree, you see the trait in generation after generation. The risk for each child of an affected person is 50%.
But hang on, what about those cases you sometimes hear about them, where a child has a known AD condition, but neither parent has it? That seems to break the vertical rule.
Good point. That brings in a really important concept: the de novo mutation. This means a new mutation happens spontaneously, either in the egg or sperm that formed the child, or very early in embryonic development. It wasn't inherited. So the child is the first person in the family with that mutation.
So it just pops up randomly?
Sort of, it's a spontaneous genetic error. And interestingly, the chance of some de novo mutations happening seems to go up with the father's age.
Okay, moving from autosomal to the sex chromosomes. X-linked patterns. Let's start with X-linked recessive, XLR, mutations on the X chromosome.
Right. And because males only have one X, they're XY, while females have two, XX, these conditions affect males much more often. A female carrier, one normal X, one mutated X, is usually unaffected, but a male only needs that one mutated X to show the disease.
And a key thing to look for on the pedigree is no male-to-male transmission, right? Because dads pass their Y chromosome to sons, not their X.
Exactly. No male-to-male transmission is a big flag for X-linked inheritance. Also, daughters of affected males must be carriers because they get his only X chromosome.
Then there's X-linked dominant, XLD.
Yeah.
Still on the X, but dominant.
Yes. This affects both males and females, although it can often be more severe in males. Here's a key pattern. An affected father will pass the condition to all of his daughters because they all get his X, but none of his sons, they get his Y.
Okay.
And there's another important detail for some XLD conditions. They can be so severe that they're lethal in males before birth. So you might see families with affected females, but fewer live male births than expected.
Wow, okay. And the last one, the outlier, mitochondrial inheritance.
Yeah, this one's totally different. It involves mutations in the DNA found inside mitochondria, those little energy factories in our cells, outside the main nucleus. And here's the unique rule. Mitochondria, and thus their DNA, are passed down only from the mother to all her children, sons and daughters alike.
Fathers don't pass on mitochondrial DNA at all.
Nope. Comes exclusively through the egg cell. So you'll see transmission from a mother to all her offspring, but an affected father doesn't pass it on. And because mitochondria are about energy, these diseases often hit high-energy tissues, hard brain, muscles, eyes.
Okay, that's a lot of patterns. Can we quickly summarize the key flags to tell them apart on a pedigree?
Sure. Big ones. See male-to-male transmission. It can't be X-linked or mitochondrial. It must be autosomal, either dominant or recessive. Does the trait seem to skip generations? That strongly suggests a recessive pattern, either AR or XLR. Does it appear in every generation? Think dominant, AD, or XLD.
Recognizing the pattern is step one. Step two, and often the most critical for families, is calculating the actual numerical risk. And we need to remember, this is always about probability, right? Not a guarantee.
Absolutely. It's an estimate of likelihood. Now, for the simplest case, like 2 known carriers for an AR disease, AA, the risk is that straightforward 25% or 1 in 4 for each pregnancy. But reality is usually messier. We often don't know everyone's exact genotype.
Right. So let's tackle that complex calculation example from the sources. It involves 2 cousins, labeled 3.1 and 3.2, who are worried about having a child with an AR disease that runs in their family.
Okay, first step is always to anchor what we do know. We look back at Generation I. We see individuals I1 and I2 had an affected child. TI1. For an AR disease, that affected child must be A.
Which means both parents, I1 and I2, must be carriers, right? They have to be A to each contribute an A.
Exactly. So we've established I1 and I2 are AA. Now, the tricky part comes with our unaffected child, individual I2. This person is the parent of one of the cousins we're interested in.
Okay, so II is unaffected. What are the possible genotypes?
Well, the standard AA-XA cross gives 14AA, 12AA, and 14A. But we know IA2 is not affected, so we can eliminate the A possibility.
Okay, so we're only looking at the AA and AA possibilities that result in an unaffected person.
Precisely. Out of those three remaining possibilities, AA, AA, two of them are the carrier state, AA. So the probability that BA2, this unaffected sibling of an affected person, is actually a carrier isn't 12. It's 23.
Wait, say that again. Because they are unaffected, their chance of being a carrier goes up from the standard 12 to 23?
Yes, because we excluded one of the four possibilities, the affected A state. We're adjusting the probability based on the information we have, their phenotype, the fact they are not sick. This is a really critical point in pedigree analysis. It often surprises people.
Okay, that is a deep dive moment. Using the fact they're healthy changes the odds. So A2 has a 23 chance of being a carrier. How does that flow down to the cousins, the 1 and 3, 2?
Right. So the calculation then depends on the relationship between the cousins and I2. For this specific example laid out in the notes, based on how 3-1 and 3-2 are related back to I2 and the original grandparents, the probability that each cousin, 3-1 and 3-2, inherited that A allele and is therefore a carrier is given as 12.
Okay, so we need T2 to be a carrier, 23 chance, and 3-1 needs to inherit the A, 12 chance, and 3-2 needs to inherit the A, 12 chance.
Correct. And if both 3-1 and 3-2 are carriers, then the chance of them having an affected A child together is the standard 14.
So we just multiply all those probabilities together.
Exactly. It's the multiplication rule. The probability that I2 is a carrier, 23, times the probability 3-1 is a carrier, 12, times the probability 3-2 is a carrier, 12, times the probability their child is affected if they're both carriers, 14.
Okay, let me do the math. 23, 12, 12, 14, that's 2 over. 3 times 2 is 6, times 2 is 12, times 4 is 48, so 248.
Yep, 248, which simplifies down to 124. So the risk for their child having the disease based on this pedigree information is 1 in 24. Much lower than the initial 14 if we knew they were carriers, but much higher than the general population risk.
Wow. Okay, so in this one deep dive, we've gone from Mendel's peas, way back when, to diagnosing a specific condition like CF, involving compound heterozygosity, understanding inheritance patterns, and then doing this quite complex probability calculation to get a specific risk like 124.
That's the power of it, really. Putting all these pieces together, the language, the patterns, the math, lets us move from general ideas to very precise, potentially life-changing information for families. Understanding zygosity, reading pedigrees, these aren't just academic exercises.
Absolutely. So here's something to think about as we wrap up. We saw how knowing someone is unaffected changes their carrier risk, like IA2 having that 23 probability. Now consider one of those cousins, say third one. What if they have modern genetic testing, a specific test for that family's mutations, and the result came back negative, definitively showing they don't carry the mutation? How would that single piece of new information totally change that 124 risk calculation for their potential child? It really shows how dynamic risk assessment is, always updating with new data. Something to ponder.